Sorry? Okay, all right. This was the last slide, right? This one? This one? This one? Okay, all right. So anyway, we're going to continue with the first basic lecture. And this was probably the last slide I talked about. And remember, there was few terms that I said that I will use. In the last lecture, I talked about innate immunity and adaptive immunity. So these are the two terms that I want you to know exactly what are the differences. The other term that I used was, again, innate immunity or acquired immunity. So acquired and adaptive pretty much are the same. Okay? And I also said that innate immunity is a hardwired immunity, so everybody is born with that type of immune system. And adaptive immunity is something you adapt to the environment, because the environment that we live in, we are continuously being exposed to different antigens. And I also said that human bodies have a mechanism where they can differentiate between self versus non-self. So that is the basic information that our human system has. In order for us to mount an immune response, we want to make sure that we identify and recognize something that we consider as non-self. Okay? So these were some of the terminology that I used. And then again, this slide basically gave you an example of an acquired immunity, meaning that we deliberately expose you to a possible threat that you may see in future. I gave an example like a flu shot. So this is based upon this assumption that So this is based upon the assumption that human bodies have to draw a line. What is it that I want to react to? What is it? Because the immune system basically has to decide what is it that it wants to react to? What is it that it wants to mount an immune response towards? And that has to be precisely regulated. If it's not regulated, then we are in trouble. And that's what we normally see, that we have allergies. We have allergies to almost many things. That we have food allergies, we have skin allergies, we have allergies like asthma itself is a form of allergy. So our immune system in this case has reacted, which is good, but reacted in a way that possibly is not healthy. That possibly is deleterious, and that possibly is more exaggerated response. So we need to understand what is normal versus what is abnormal so we can pretty much differentiate. Okay? Now, in vaccination, for example, we take an antigen from a virus and inject you with this antigen. So this antigen your system has not seen before. And the body mounts a protective immune response. And the reason is that should body be exposed to that antigen ever, body is prepared. It's just like, there is a list over there, the body identifies that if such and such virus comes with such and such pattern, we need to respond. Okay? And sometimes what we do is that, for example, you already have an infection. So you already have an existing infection, your body is fighting an infection. And in this case, we can't give you antigen because you already been given an antigen because you are infected. So now we want to help the immune system. Your immune system is trying its best, but sometimes it doesn't really build up to enough strength to fight that antigen. So in that case, then we have to give you passive immunization. So we give you prepared material like antibodies and many different things. So that will be considered as a passive immunization. And then finally, adaptive immunization is that because immune system is based upon cell responses, especially in cancer, transplantation, and so on and so forth. And these things, obviously, we will consider in detail when we take them individually in individual chapters. So we take cells out of a cancer patient and prepare them to fight against cancer and inject them back into the cancer patient. So this type of thing we call adaptive immunization. No. What I said that if your body is exposed to an antigen and it builds up an immune response to an antigen, is active immunization. If we do not give an antigen to the body, because body may have been exposed to antigen already, we give some other thing like antibodies to help fight that antigen. That will be considered as a passive immunization. Right? And the third thing is cells transfer from one person to another person. So anyway, these are some of the characteristics that we already discussed and we'll discuss in detail again. You can see that acquired, of course, immune response is very specific. So if you are given, sometime you hear in the news that the flu shot that was given was not very helpful. The reason being that viruses mutate. So they basically make that vaccine ahead of time, assuming that if we give an antigen like ABCD, so that antigen will be shared by the fresh virus. Sometimes it does not share that, so it mutates. So the vaccine is then infected. So it's very specific. And then again, it's very adaptive. As the word goes, it has to adapt to a particular. For example, you heard Dr. Coleman mentioning that you have to have hepatitis B vaccine, you have to have polio, you have to have, measles, so on and so forth. So what it means is that all of these are antigens that your body needs to figure out, understand, and mount a response towards. Right? So that's what adaptiveness means. And then again, the whole idea is that body would only react when body would think that this is a non-self. It is a non-self. So if it's non-self, if the body considers something as self, it would not respond. Right? And then again, body also has a memory. So it's just like if you open up a program on your computer, and then again, either computer, if you put a password, say, do you want system to remember your password, or you want the system to at least, get the cookies from that server. So that kind of information exists in the immune system, whereby immune system would remember that particular sequence of antigens. And next time, if the immune system is exposed to that particular antigen, it is already prepared ahead of time. So that's what the memory cell is. And then again, we'll talk about the cellular and molecular basis of immune response, and basically, whole book deals with that. So the whole book of immunology deals with that. As I said earlier, in immune system, we have cellular components, and we have cells involved in immune response. And again, if you remember me saying that before the advent of all these molecular techniques, we were not sure what kind of cells are we looking at. But of course, you can see that all the way from 1950s, 1994, when microscope came in, electron microscope came in, many things were there, that gave an idea as to what type of cells are there. So for example, lymphocytes. We talk of lymphocytes. Now we know there are different types of lymphocytes. They are B cell, T cell. And these are the cells we'll discuss in detail when we talk of immune system. So this first lecture basically just gives you the names of the players in immune response and what is it that is going to help us in immune response so we can make out the difference or we can go and help our immune system to build up its defenses.
What does ABC stand for?
Antigen presenting cell. Okay, and major histocompatibility cell. These all are abbreviation given in your book. I'm just giving you an overview for that. We'll discuss that in detail. And then there are interactions between different cells. So an antigen presenting cell is going to interact with T cells. So this is what you will see. And then again, some of the cells basically you will see in both innate and acquired immunity. We talked about inflammation. I'll talk about inflammation again, and this time I will emphasize on the cell like neutrophils, mast cells, and many other cells. So a part of your paired learning is that if you can make two columns, both of you can write cells of innate immunity, and then you can write cells of acquired immunity, so you can differentiate. So what are the cells that are primarily for innate immune response and what are for acquired immune response, and what are the cells that may function in both. Okay. Now, there are many different theories, and you can see that this is a basic immune theory being propagated in early 1950, is that what is it that an immune system entails? So if you see an antigen, what happens? For example, this is a layman knowledge. Everybody will say, you know, I took the flu shot, it was very helpful, and I'm going to take it again. So the layman probably doesn't know exactly how it works. Now, one of the objectives of this course is that you, as a pharmacist, should know why and how it works. Okay? But before we do that, we need to know how our normal immune system works. What are the cells there? What are the tissues there, right? And then again, these are some of the scientific hypotheses based upon good observation. So if you remember, all tissues are composed of cells, and cells come from stem cells. And stem cells then divide into undifferentiated cells. So these are cells, for example, there are two cells. I mean, this is like, I don't want to teach you reproductive immunology, but these are like male and female, and it starts from there. And then we have very specific stem cells. Now, these stem cells will differentiate into muscle cell, cardiac cell, liver cell, kidney cell, so on and so forth, along the line of development. Likewise, they also differentiate into immune cells. So that's what we'll take in this part of the lecture. But also remember the major thing, self versus a non-self. So when a stem cell is dividing into daughter cells, so each one of the cell, like a big store, each one of the item will have that computer ID. It has a barcode there, right? And if you look at the barcode, you will not be able to understand. We either have to have an app to read that barcode, or it's going to go to that till, and it's going to run through, and they'll give you the details on that. So any cell that a stem cell divides into carries a barcode. And there's a genetic barcode, and we're going to discuss that in detail. And the reason is, for example, you know, I bought an iPhone from Best Buy, for example, and say I've lost the receipt, I want to return it. You're going to run the barcode and say, well, our record shows that you did never buy it from Best Buy. You must have bought it from T-Mobile, because all this information is there in the system on the barcode. So our immune system gives a tag to each and every cell of our body. And there are scanners in our body that will be continuously scanning it. I mean, I can tell you that there is one cell that runs in your system within 3 days, and this is the job of this cell to make sure that you are what you are. So you are self. So it is not ready to appreciate anything which is non-self. Right? But again, things can go wrong. It's biology. So when things go wrong, that's where the disease comes. So I'll touch upon some of the diseases in the first lecture just to emphasize upon you. But of course, the clinical immunology, the last 10 lectures are designed for that. But I just want you to understand that things can go wrong when your own cell is unable to pick up whether you are self versus non-self. And guess what happens? It's going to pick your cell up and react to it. It's called autoreactivity. Okay? And of course, it's going to lead to autoimmune diseases. Right? But before going in detail, remember that these are lymphocytes. And there are different types of lymphocytes. I will discuss that in detail. But for now, just remember 2 types of lymphocytes, T and B cells. Then again, when I discuss T and B cell, 2 of you, one will act as a T cell, another like a B cell, and then you can start writing what are the differences between B and T cells. Both are lymphocytes. They look alike. If you see under the microscope, they look alike, but they are not. And every other day, they are coming up with new differentiation. And also remember, they are cancers. They are cancers of B cell. They are cancers for T cell. Okay? So BCR stands for B cell receptor, T cell receptor. The whole idea is that, remember I said there's a barcode. But that barcode, if you look at any barcode, it's not only one bar. There are tons of bar there. I don't know how many, 50, 20, 30, depends. Some are long, but I would say most of the time, maybe like 3 inches long. But to an ordinary eye, they look alike, but there is an information there. So that information, basically, we need to discuss. And if you go in detail, this is a cell, and it has receptors there. So these are the receptors. And the other word that I'm going to use is epitopes. Epitopes, basically, again, are very precise locations of those receptors, like sequence of amino acids that I will discuss in detail in immunology. Okay? So this gives you a rough example of what I just said. You have a stem cell. It's going to divide into uncommitted cells, proliferation, and many different cells. One of the cells that this stem cell will form are lymphocytes. And lymphocytes, basically, the basic structure is they have narrow rim of cytoplasm with a big nucleus. And any cell which has a big nucleus, just keep in mind that it has a lot of nuclear events. And then it starts expressing those receptors. They are receptors on top of that. It's just like hair on your body. You know, there are different tiny little receptors, and they are all very important because our drugs are going to block those receptors. And these are identification points. Now, while these cells are being differentiated, you can see some of the cells have this capability because it is a genetic differentiation. It's a random kind of production to begin with. And you give a sequence to each one of them. There's always a chance that you make an error. And if an error is made at the beginning where you are making those cells, there are chances that these cells will go and identify you as a self, right? And then kill your own self. But also keep in mind that there are two different events. One events are happening in intrauterine life when the first batch of lymphocytes are made, and then also now your bone marrow is producing it continuously. It's a half-life of these cells. Half-life of red blood cell 4 months. Lymphocyte, even they say variable. Some of the lymphocyte can live forever, like memory cell. You don't want to kill your memory cell. But recently they have noticed that sometime you lose memory cell as well. Okay? Now you can see from here, then cells are removed. So there is a mechanism in our body in the intrauterine life where there are checkposts in the body and want to make sure that the cells that carry weapons of destruction, so to speak, because these cells have this capability to kill. They are killer cells. There are cells in our body which are killer cells by the design of it. And some of them will kill anything that comes nearby. But the other things would wait and want to make sure that they recognize you as non-self before they will kill you. And that happens in different diseases, but you can see they are removed, and the bunch of cells that are left are not going to identify you as a non-self, and then again, they will just recognize an antigen. Just like you want to go and apply for a cell phone number, right? And you say, I want to have such and such number. So they say, okay, let me check if that number is available or not. Right? So the idea is they don't want to give two people the same number. Now, when an antigen comes, remember I'm just saying antigen, so it's a protein. It could be a lipid. It could be a carbohydrate. So it comes with a sequence of amino acids. I'm not talking about that. I'm just giving you an overview for that. So it looks at it and identifies it and prepares a defense against that. So that kind of defense basically to any sequence of antigens is simple words, antibody. So antibody is a response towards an antigen. Okay? And then again, there are some terms that I will use as we go along. So we had innate immune system, acquired immune system, adaptive immune system, immunity. Then we talked about now two important limbs. We call two important limbs of immune system. So the two important limbs or hands of immune system is, one is a humoral or humoral, and the other one is cellular. So the whole response is with both hands. So humoral is the one that is an antibody mediated, and the cellular, of course, is a cell mediated. And also remember that cell is a cell and antibody is not a cell. Antibody is a protein. It's an immunoglobulin. We'll discuss that in detail. It's an immunoglobulin. And then there are many other things we'll discuss again, and you've already studied. Complement system is going to come again. We have full lecture on that, and we have neutrophils, polymorpho-neutrophils, and many other things. But for this part of the lecture, I think this is how an antibody looks like. And there are two heavy chains, two light chains. We'll discuss that that's why I said this is humoral immunity, because it's not cell-mediated immunity. So this is not a cell. Okay, this figure, basically the prior slide is explanation of that. So B and T cells, very rough cartoon of B and T cells. They're both lymphocytes, they look alike. If you were to look at an ordinary microscope, you would not be able to differentiate. You can only differentiate depending upon the receptors that they have. It's just the same receptor that you saw in endocrine system. Exactly the same kind of an idea. And also keep in mind, like in drugs of endocrine system, the drugs that we are going to use is again either going to, yeah.
Earlier you were talking about nuclear events. What are those?
Well, we discussed that in detail when we go to translation. I just wanted to inform you, any cell which has a big nucleus means that it needs a nucleus. There are many things that are happening within the nucleus. That's why it's a big nucleus there. Transcription, translation, all these things we'll discuss in detail. So you can see, this is a B cell, this is a T cell. B cell has a receptor, right? And remember, there are tons of receptors. I think I'll let you know which are the receptors that we normally use to block in terms of drugs, so you have an idea. And then there are some of the receptors, they are again very visible, obvious moieties, so you can see them. Right? For example, in my lab upstairs, when we separate B and T cells, we identify all these markers. So we have ways to do that. So B cell has a marker which is basically an antibody. So that's interesting because antibody is normally secreted, but sometimes you see antibody attaches itself on top of a B cell. And if I was to ask you what is the difference between a B and T cell based upon this figure, you will see the T cell does not have an antibody attached to it. T cell has another thing which is called T cell receptor. And 50% of drugs that we have right now for cancer therapy are going to target that. And we will discuss that all the way to the amino acid sequence. So we're going to go in very detail for all these, so it's just an overview for that. Okay, then again, functions of various T cell. When I say T cell, so there are many different kinds of T cells. Depending upon their function, some of the T cells help, we call helper T cell. We write them TH. Some of them are cytotoxic, they are killer, we call TC. Some of them produce cytokines, which are secreted proteins. So depending upon what their function is, we give them a name. And that we will discuss as we go along. The other important thing that you keep in mind is that this immune response, IR stands for immune response, basically is based upon your genes. It's not random. Like, for example, you say, why do you have allergy? Well, my mom had allergy. So something you feel that something is passed from the genetic material. Normally, every one of us will carry 50% of genetic material from the father and 50% from mother. The chances are that you are going to inherit whatever they had. Cancer, allergy, and many more things. But we need to understand how does the body do it. So the idea is that we have this capability, it's just like immune system has this capability to generate those IDs, to generate, as I said, give an example, a telephone number, or generate that barcode for each and everything that is out there in the environment. It may sound to you odd, but just like alphabets, 26 alphabets, so everything which has been written or there in the libraries or in the press every day is basically coming from those 26 alphabets of English and so on and so forth. So we have very specific number of cells that are there to help us do it. Okay? And that again is very specific. That's the beauty of immune system, is that it normally should not miss. It will make sure that it gives you an ID that this is your cell. Okay? And then again, it was basically discovered by one Japanese, Tonigawa, and one other American. They basically found out what are the genes associated with that, because that's interesting and it's important as well. Right? So that we react. Some of us may have a peanut allergy. Some of us may have allergy to very specific thing. So that information basically is running in our genes. And we need to know what those genes are. Okay? But you can see that we have this capability of generating that. So if you expose 10 million antigen to our immune system at one time, it will recognize that. Is that specific? So it has this capability. I don't know, many of you know that, especially for us, we just put like Google search. And those who are interested, that Google Excher servers are maybe more than a couple of football field sizes. They are in California, right? They're all sitting over there, and they are dealing with that. So you have to have that physical structure. So interestingly, we human beings have it. So what are the practical implications of what we are going to study? For example, we talked of vaccine. You can see vaccines are mandatory. They're not going to even allow you to sit in an exam if you're not vaccinated. So it's there, proven. And then again, AIDS, cancer, everywhere we are talking about this immune system. And some of the diseases, if the vaccines were not developed, would have totally exterminated the mankind. They still do think, if God forbid, we have in swine flu or things of that nature, the expected mortality will be in millions. So we have to prepare ourselves. So there are many things on Develop 9. We're talking about vaccination. We're talking about immunoprophylaxis. That's what you normally do for all these things. And then again, there are many things happening in terms of treatment of, for example, in reproductive immunology, that to treat some of the infertile couples, and even for contraceptions, they are coming up with some of the immune drugs. They are there, and they are being used in mammals, human, cats, and so. Because remember, this is not a human population they want to control. They also want to control population of other animals as well. Okay, and I did talk about the damaging effect of immune response. We'll talk about that in detail as we move along. Okay, and let me jump to the next one, because we are already running one lecture behind. Okay, now remember I said there is a system over there. The immune system is geared towards picking up an antigen, right? You pretty much got that, right? So anything which is considered by the immune system as non-self, it will mount an immune response to, right? So we already know what an innate and acquired immunity is. So I'm not going to spend much time on that. You can read it on your own. I will spend some time on some of the organs and cells which are there, and then the question is how an antigen enters us and what do we do, how an immune system will recognize that. So that is something to talk about. Okay? Now, when we say innate immunity, basically what we are saying is that this is a non-specific immunity. Your skin and mucous membrane should protect you from all microorganisms. Your skin should not react to the clothes that you wear. Your mucous membrane in the lung should not respond to every antigen that you breathe in the air. Your mucous membrane of gastrointestinal tract should not react to every food that you take. This is a part of innate immune response. So that's something that we have to keep in mind. And then there are general processes the body has of something which is foreign. Body will go, there are specialized cells which are called phagocytes. They will go and kill it. Because the whole idea is that this is a defense system. This is the defense system in the body where you do see, for example, again, we have stem cell, and you can see that all the cells which are there in blood, these cells basically have a function. So they have a very specific function. If they have a function which is a non-specific function, what it means is that they are going to react against every microorganism regardless of whatever that microorganism is. For example, if you get an infection of flu and you have not been vaccinated, so your body is going to respond to it. but it's going to respond in a way that it can possibly do so. It is not prepared. It's going to be like random. It's not prepared for that. It's just all of a sudden, a panic is created and it's going to go after that. But you can see these are some of the developmental pathways that you have in bone marrow. And you can see from here, most of the cells that we have are myeloid progenitor cells. So these are the myeloid series of cells that we normally use: neutrophils, basophils, eosinophils, monocytes, lymphocytes. So these cells are normally, when you look at infection, you do look at the counts of the cell. So you look at these counts. So these cells are a part of the immune system, but they basically are non-specific. They are not going to recognize anything. They would just do what they need to do. Neutrophils are filled with a lot of reactive oxygen species killing material. But the moment neutrophil is going to phagocytose or eat up a microorganism, it's going to kill it. And these tissues, basically, these cells are considered as tissues. That's why when we say blood typing, we say tissue typing. So these are tissues. On the other hand, when you have T cell and B cell, you can see this is where the immune cells are coming from. So immune cells, T cells are made in thymus, in intrauterine life. Most of us have thymus in the middle. As we come to this world, this thymus involutes, it disappears. So the job of the thymus is then taken up by bone marrow. So for all practical purposes, bone marrow will produce B and T cells. And you can see from here, there are T cell precursor cells and B cells. And initially they were thymus. But remember, having said that, sometime the thymus may persist. So that's again is abnormality. We discussed that as a disease. And then again, there are some parts of immune system that are there that will help us to mount an immune response. This is just a general slide that you can see that the processes that some of the cells have is that anything that they see is foreign, they will engulf that. And it is the same process that these cells have for acquiring food as well. So remember, macrophages, they are cells, they need food, so they are going to get, just like glucose. So they need glucose for oxidative phosphorylation, so they were going to take it in. And then again, pinocytosis, all those diffusion mechanisms occur, and they are non-specific, and they can be a part of that. And most of the cells of immune system and innate immune system carry bags of lysosomes or lysosome type material. They are nothing but weapons that cells have to digest or kill or remove. So these are some of the things that normally happens, because remember when I told you that the cells are replaced over time, so what happens? So these cells have to be physically taken away from our system. So this is the mechanism that we normally do that. Now again, we already discussed that, cells involved in innate immune system, neutrophils, macrophages, and then there are very specific cells of immune system that lie in different parts of our body. There are some immune cells, they are there in the blood, so they are circulating. And there are some immune cells, they get lodged in some parts of our body. In this case, you can see Kupffer cells. They are usually there in the liver. We have alveolar cells in the lungs. We have splenic cells in the spleen. And we have peritoneal cells in peritoneum. And we have glial cells in central nervous system. There are tons of cells. And these cells are important because they are going to play a part. And they are strategically placed in tissue where there is a chance that they are going to encounter an antigen. So they are there to pick it up. Then again, the largest organ in the body is skin. So we have dendritic cell in the skin. Our skins are full. That's why if you go for allergy testing, And that allergy testing, basically, they're going to do on skin. This means the skin has those specific immune cells, like Langerhans type of cells. Okay? And then there's some of the cells are there circulating in the blood, and we call natural killer cells. So these are naturally killing cells. The moment they see something non-self, they will kill it. But some of them are specific T cells. We call natural killer T cells. And remember that these cells are important because if your body doesn't have this capability to generate these type of cells, then the problem is that if you, God forbid, if you have a cancer, then the cancer is going to flourish and grow because there is no stop to that cancer. And this is a picture of neutrophil. You can see polymorphonuclear leukocyte, and you can see a typical structure. And the whole idea is that, like any cells, these cells have a function, and based upon the morphology. This is a picture of a scanning electron micrograph of a microvilli. So you can see from here is a macrophage. So there is a macrophage, which is a cell. And normally, if you were to draw a macrophage, you will draw like a circle. That's what we normally do. But the fact is that it's not circle, it is ruffled. So all those things which are jutting out of these are very specific receptors. And these receptors are there, which are going to be discussed in detail in immune system. This slide basically will give you an idea of some of the cells that I told you that have the ability to defend us. So they are there to defend us. And the moment they are there, they see something going wrong, something which they consider as non-self, they will actually go and kill it, but keep this in mind without identification. They are just going to arrest you or shoot at you without asking for anything. The only thing they need to know that they have a way of finding out that you are non-self. So in this case, you can see this is a typical structure of a killer cell, right? And this is a normal cell. Remember I said, and just keep in mind that our skins are continuously being rejuvenated. So skin's cells are going through rejuvenation. The mucosal cells, they are cells which are continuously being, produced and proliferated. But the dead cells are removed from the body. So how the dead cells are removed from the body is that, for example, if there is a normal cell and your gene, that again is your gene, will come and tag it. So that's another thing that you have to keep in mind, that no cell will be removed from the body for all practical purposes if your genes don't come and look at it. So your genes will come, look at it, and they will put a tag. So that genetic tag that we'll discuss in detail in one of the full lectures, basically different types. In this case, you have is called major histocompatibility complex, MHC. So when you go, you want to donate blood or you want to donate tissue, they say do blood typing. So when they do blood typing, they are testing your gene sequence for major histocompatibility complex, which is MHC class 1. So most of the cells on your body would express that. And the killer cell, when it comes to your normal tissues, if the killer cell looks at your MHC, there is a message in the cell that will say no. So this would not allow the killer cell to kill your own tissue because now it knows it's normal. Does that make sense? So that information is there. And remember, this is a unique information for us to understand. For example, if there is a cell, and this cell, like this case, doesn't have your MSC receptor attached to it, right? So this means there is nothing to stop inhibition. So the killer cell will come and it finds there is either decrease or absent MSC class 1 expression. So it will kill it. And that basically is the, many times you saw, and some of the, in virus diseases, like if you have herpes of the lips, so you see a lot of tissue destruction. Simple example, I mean, virus causes a lot of damage, tissue damage. So who is doing this damage? Virus? No, your own cell. Because what virus would do is, that virus would not allow the MSC class 1 to be expressed on your normal cells. So that is how virus operates. So if it's not expressed, then your cells will go and kill it. This is bad for us. It's a bad news for us. Okay? This is bad news for us. Okay? But remember that anything which is bad, we can get a good lesson from that. So once we know that this is what virus does, and if we were to, just imagine just like last point, and then I'll stop. So we know that virus causes that problem, right? And this is bad for us. So we need to devise a drug that would stop virus doing it. So we have to do something with the receptor. The other good thing is that if in cancer, because remember it says virus infected or tumor cell. If the tumor cell does not express that MSC class 1 antigen, the tumor cell will be killed. And we want to do that. We want to kill the tumor cell. So what we normally do is that we will devise of drugs that will remove the MSC class 1 receptor on top of the cancer cell so that our cells will come identify and kill it. So most of the immunopharmacology or cancer therapy or receptor therapy is based upon that notion. So you understand what's going wrong, and then you can adjust that wrong and correct it. Okay? So I'll stop here, and we will continue at a couple of announcements. Where is the sheet? Where is the sheet for the pairing sheet? Okay. You know, yesterday the sleeping, apart from a couple of the student housekeeping thing, I said no eating, I still see people eating. I said no sleeping, people still sleeping, that's fine. But remember, attendance is not mandatory. So if you're really sleepy, you want to eat, you can go out and eat and sleep and come back. But what I want you to do, because the body language tells me, like, oh, why is it, why we're going in so much detail? So you will feel that in immunology if you don't read ahead of time. It will sound French to you. And if it's that difficult in the class, it's going to be very difficult for the exam. And I'm just warning you ahead of time as well that in microbiology, people fail the course because they fail in immunology. So please make sure that you can start reading ahead of me and try to understand this concept. Because if you don't read on time, by the end of the exam, you would not be able to keep up with the speed that we're going to go to. Okay? All right, thank you.